Hi, my name is Chloe Blanc, and this is the video for the Week 2 lab bacterial enumeration. Today is July 15th. It is around 6:40 p.m. and I just want to go over the results that we had that we started on July 11th. Around 5 is when we started this process. So the process began was after creating the agar plates. I had 6 tubes, each labeled 10 minus 1, 10 minus 2, all the way to 6, and with corresponding agar plates with the same number system. In each tube, I had to put 2.25 milliliters of water and mix it with 0.25 milliliters of yeast, which is the S-serum base that we've been using before in the prior experiment. I then had to dilute it from the first tube, the 25 milliliters from the first tube into the second tube, and then so on and so forth. So go from the second tube to the third tube, all the way to the last one. I then had to take 4 drops from each tube into the corresponding numbers that are on the agar plates. And on Friday I checked, that was the 48-hour period mark, wasn't happy with the growth results. Checked on Saturday, wasn't happy. Today's Sunday, the last day, I decided to evaluate what has happened. So with each plate, there's very little colony growth. The most I had was 15 because they either clumped into big groups or they're very little tiny dots. So the original number was 15. The least I had was 6, and that was because there was just big groups of the colonies. So when I calculated the CFU, I calculated the smallest growth number, which was on plate number 5. I didn't think it really mattered at this point because each one of them had such small growth, and that was about 6 million CFUs per milliliter. So I'm not very happy with the outcome of the poor growth, and there could be different things that were involved. On the first one, there was some fungal contamination, so it could have been a process of the inoculation loop had gotten contaminated and I mixed it onto the first one. It could be a number of different things. It could also be that we are struggling with heat over in our house right now, so we do not have perfect temperature for the incubation. So this just shows that as scientists, you have to repeat experiments multiple times if you have a hypothesis that you want to test. Again, I'm not very happy with this result, but I'm glad to hear from the professor that the growth, I am seeing growth, it's just not enough that what we wanted, the goal was that matched the instructions for the 30 to 300 CFU goal. But this gives me hope for the next experiment that hopefully I have better aseptic techniques and to limit the contamination process as possible.